thank you very much for coming back, and let's start our last session of the day. I would like to ask Gábor Tari to give his talk. He holds master's degrees in geophysics and geology from Eötvös University in Budapest. He graduated from Rice University with a PhD in geology and geophysics, and he will provide insights into the current state of natural hydrogen exploration. The title of his talk is Natural Hydrogen Exploration, Quo Vadis.
Thank you very much for this nice introduction. It's a great pleasure to be here, and I really appreciate the organizers to have me as the only representation of by the Earth Science department. So this rock is a very special rock. I have it right here, and in fact, if you are into rocks, I'm more than happy to give it to your hands and then have a closer view. The story about this rock is that it's one of those special rocks, as we found out, which can generate and did generate hydrogen, natural hydrogen. And this is really the story I'm going to talk about. Let's see how this works. All right, so the agenda in very crude terms. I will talk about hydrogen, what natural hydrogen is in the spectrum of all the other hydrogen species you keep hearing about on radio, on TV, and whatever. It's an important place to emphasize that this is the only hydrogen species which is made by Mother Nature. All the other ones are artificial, if you will. I'll show you something very exotic. I'll take you on a short side trip to Mali, West Africa, where there is the very first natural hydrogen field on this planet. And it's kind of an important observation and fact we have to dwell on a little bit. And I'm coming from the oil and gas business, 30 plus years in the energy business, as they call it these days. And the question for us is that the hydrogen, the natural hydrogen, if we go after it, and we are trying to go after it, is it going to be very similar what we have seen in the oil and gas business? Are we going to have the new energy source coming from the ground, just like what we had with oil and gas for so many years, more than a century? All right, and I will conclude with some general observations. So about energy in general, we all can appreciate the fact that everywhere we consume more and more energy, and this is a good visual to see how incredibly addicted we are to energy. I wish I would have an image, the same size, or same style, 2025, but we all agree it didn't get any darker. If anything, it's getting lighter and lighter. And of course, many people mention AI. The big thing about AI, that it sucks up so much energy to run those computers and everything, that if anything, the energy need is an exponential curve which is kind of running away from us. So in that context, we really need to think about what we're going to do with our energy need going down this road. And this is one assumption, how the global energy mix will look like in 2050, if things will work out just fine, quote unquote, if we more or less try to follow the Paris Agreement, if we do that collectively. And I don't have to emphasize the fact many of you, just like myself, worked in the US and still work in the US, and then, for example, the establishment getting out of the Paris Agreement will put a twist on the storyline. But if you just focus on these numbers, a couple of things to emphasize: the oil and gas as a major energy source will be still there. I wish I could tell you that we're going to get rid of it, but we won't be able to. We will have to have a certain amount of oil and gas to produce energy. Another interesting observation, that nuclear will go up. Countries like Germany will have to take a U-turn on their policy. This is unavoidable. Nuclear will come back. Hydroelectric will stay the same because there are just so many dams we can build on our rivers and lakes and everything. It's going to stay the same. Coal is an ST operator. That's dropping down. It has to drop down significantly. But of course, it's hard to tell to scale back on coal consumption to countries like China or Poland on this and the other. So it's a little bit of a geopolitical challenge. But on the left-hand side of this figure, you can see that this big package of renewables, 42% altogether, has very interesting elements to it. And there are no numbers on the individual items in there. But the dark horse is really the hydrogen. and I put a question mark on it. And in this hydrogen bucket, as I'll show you in the next slide, there are a lot of things. And natural hydrogen is going to be in this dark horse category, as you will see. Now, the colors of hydrogen, it's the rainbow colors. It's politically very correct or incorrect. If you want to look at it, depends on where you are. But the takeaway is that many of these hydrogen species you already heard about, like green hydrogen. You take the water molecule, you split it using electricity. It's a very straightforward thing, except that you need to have a lot of energy to break that bond. But then you have hydrogen and it's all nice and all clean. You have to think about where the electricity comes from. And there are some other much dirtier versions like the brown and the gray and even the black hydrogen, which comes, for example, out of coal. And then you end up producing a lot of CO2 in the process. So it's really not something you want to do because of the carbon footprint of your process. But here at the bottom, there are two categories. The gold, or people call it the white hydrogen, what I'm going to keep referring to as the natural hydrogen. This is the one Mother Nature fixed for us. It's already in the ground. And orange, which we're not going to talk about today, this is something which you generate or induce within the subsurface. It's a little bit like shale or shale gas. It's a different category. So another way of looking at this hydrogen species is this. Again, all of them are man-made or artificial, if you will, except the white hydrogen, which I show on the right-hand side, where you have some sort of hydrogen accumulation in the subsurface. And the question whether you can get to it and how you're going to get to it, and is going to be economically feasible to produce that subsurface hydrogen accumulation. Now, just a few years ago, when people talked about natural hydrogen as a potential energy source going forward, replacing oil and gas, there was a lot of, and there are still a lot of negative sentiments saying that we, oil and gas companies, the industry globally, we drilled almost 500,000 wells. And we didn't really see much signal in those wells, and we drilled everywhere. But this is incorrect, because we did not drill everywhere. We didn't drill on these kind of rocks, which are actually the hydrogen producers, A. And B, many of those wells, we didn't measure the presence of hydrogen. So we have a big data gap in our very big data sets. And this is one compilation. You can see many other compilations these days showing that historically we do have a lot of reporting of natural hydrogen in wells. And an interesting side note, in the former East, if you will, the former Soviet Union or the satellite blocks, there are many more reportings because in the East, there was more tendency to measure hydrogen and helium in the gases which were encountered in the subsurface. And the US, for example, is not going very strong in this regard because of the historical gap in the data set they have. Now, I promised you an exotic trip to Africa, so let me take you to this thing which I highlighted in West Africa. And this is where the story got really wild. And the story actually goes back into 1987, when people were drilling for water in this part of West Africa in Mali. And many times when you drill for something, and I can tell this on behalf of the oil and gas companies, that they did find not water, but what they did find the hole is some kind of strange gas which was hissing out from the well bore. It didn't have a smell, it didn't have a color, but it was clearly a gas which was coming out from the ground. And the drilling engineer with a cigarette in his mouth made a mistake to try to look into that thing. He survived, but he had a little bit of a scar on his face. They analyzed the gas, and it turned out to be 98% clean hydrogen. And that was a huge game changer. They didn't know what to do with it. They abandoned the find. And much later, just a few years earlier, starting in 2017, a company came back specifically to keep drilling that area to find more and more of this hydrogen. And in fact, they drilled up to 30 wells in this part of Mali, and then they established the very first natural hydrogen field on this planet. Okay, so it was really big news. Now, the problem is that it's not that just it's a tongue twister, burake bogu, but the other problem is that it's really sitting in a place where producing hydrogen wouldn't make any difference, because you are in the middle of nowhere, and as you know, poor colleagues in Mali, they have much bigger problem than energy and this and that, given the geopolitical situation. But the thing to keep in mind is that the accumulation itself has a lot of resembling elements to a gas field, which we are used to in the petroleum business. But then when you look a little bit closer, then it starts to look really bizarre. And without going into details, for example, the seal rock over this gas, which is quite shallow, is provided by these Triassic age dolerites, basalts, seals. There's no closure for the field. And when they started to produce the gas, the pressure in these shallow wells didn't decrease like what you would expect, but instead it started to increase, because you woke up the giant, kind of, sort of. It's a very different system, as I'll show you in a minute. A hydrogen system is a totally different word from we are used to, what we call the hydrocarbon system. All right. So again, I mentioned the fact that I wish this field would be somewhere in Europe, like here next to Budapest. This would be a very different story. We would be developing it, and then we would be benefiting it. In this very remote location, this is the president of Mali on the right-hand side. On the left-hand side, Professor Prinzhofer, a French colleague whom we work with on hydrogen. He's Mr. Hydrogen, in fact. And what they do with the hydrogen, they just burn it in a small turbine and then provide electricity for the field, for the little village. That's all they do, unfortunately, at this point of time. Now, I promise you kind of a comparison between a hydrocarbon system versus hydrogen systems, and it's a totally different world. And I'm not going to go into the details, but all of you appreciate the fact that when we are looking for an oil field, we expect a trap, we expect a seal, and the process which goes into filling up a oil or gas field takes millions of years, because the maturation out of source rock and blah, blah, blah. So you end up having a very static system, which is frozen in time, and we have to image it, and we have to find it by drilling and start to produce it. Now, hydrogen systems don't look like this at all. They are very dynamic, and then the filling and the migration through the rocks from a kind of a source rock system down below is a very quick-moving thing. And we are not talking about millions of years, but hundreds of years. It takes hundreds of years to make an accumulation if it stays. And in fact, this Borakabugu field that I showed you, some claim that it started to accumulate only 500 years ago. And it's in the making of being filled. So it's a very, very different world, what we are looking at. And the other important thing I'm going to emphasize is that the seepage. On the left-hand side, we barely find any evidence on the surface from a subsurface oil and gas field accumulation. In other words, the oil and natural gas cannot really get away big time and have a signal on the surface. That's not how it works. But for hydrogen systems, it's just the opposite. You have much, much bigger rate of seepage, because the hydrogen, being a very small molecule, is incredibly agile and can get through almost anything if it has time. So this is a very different situation on the right-hand side. And then the rocks, of course, which generate hydrogen, are very different rocks from the rocks which, imagine I hold up a nice source rock here, like a black shale, which would generate oil and gas. So huge differences between these systems. Now, then the question for us, for energy companies who are trying into this space for the last 3-4 years, and this is like ongoing process, it's unfolding as we speak, we would like to think that there are elements of this hydrogen hunt which could be approached just like what we do in hydrocarbon systems, and this is on the left-hand side. For example, there are beautiful salt features. Salt is a rock which can hold back even hydrogen very efficiently. And for example, in Ukraine, there are hundreds of existing gas fields in the Dnieper and Donetsk basin, which you keep hearing about every day on the news in a different context. there are a lot of recordings of having hydrogen in the gas cap of natural gas field. And it's known for many, many decades. So this is something which we know, and this works. Cool. And I'll show you this Mali example, where we have this very strange situation of dolerite seals providing the seal. So this is okay, but if we go down this road, basically the whole thing will be just like in oil business, that you find an accumulation, you drill it, you produce it, and you are done. So there is no sustainability in the whole thing. The sustainability comes from the right-hand side, where you are able to tap into a hydrogen kitchen, and then you produce it at the rate as it comes. And if you do that, then you have an infinite source. You have real sustainability. And this is the really exciting part. And I'm not here to tell you that we know how to do it. Nobody does. But this is what many people are working on in the industry and beyond. So on the right-hand side, I'll just show you one interesting thing, is the so-called fairy circles. It's a concept, it's an observation, it's a feature which we became aware, all of us, academia and industry, in just the last few years. They are very spectacular, and again, they came out of left field, if you will. So I'll spend a little bit of time on those, and I will talk about these faults, which I show on the extreme right-hand side. Faults cutting through the rocks and becoming highways for the traffic, for natural hydrogen coming to the surface. Now, on the left-hand side, if you recall, I talked about traditional traps for exploration, and this is what, for example, some French colleagues are trying to achieve in the Pyrenees, where people observed hydrogen coming to the surface. And what our French colleagues are planning to do, they haven't done that, is that they will drill wells on the scale of 20-25 million each to find accumulations of hydrogen, just like what we expect to find for a natural gas field. This is one way of approaching it. Again, to me, this is the less exciting thing, because once you find it, then you have finite resources. But this is the other category, the fairy circle stories, which show a totally different picture. And I take you back to Mali, and this is an area just east of the Buraké-Bougou hydrogen find. And what people find, and this is not just in West Africa, but in Australia, Brazil, Ukraine, Russia, and you name it, is that there are some big semicircular, very slight depressions on this planet where there is vegetation damage. The vegetation is gone or kind of subdued. And if you go in with the right equipment and measure the soil gas in 1 meter depth, you find out that there's a lot of hydrogen in the soil. Now, in the air, right here, we have half a ppm hydrogen. So these measures of a few hundred ppm are clearly anomalous. And that tells you that these fairy circles correspond to megaseeps of a hydrogen system. And this is a very important observation, which again just came about a few years ago. And then you have to start to think about, so how does it work? Can we look for analogs from our own experience being in the energy industry? And of course, one thing came to my mind anyhow, is let's look for pipeline leakage as an analog to see how that looks like. But pipeline leakage, when it comes to asking people for photos and documentation, is like dirty laundry. Nobody wants to give you anything because of the legal ramifications. No, our pipelines don't leak, not at all, no problem at all. But I could find this British PhD, which is a very cute story. Here you have a very old Victorian age 12-inch cast iron, where they run natural gas below a field of barley. And what this PhD lady found very nicely documented is that the growth of the barley is handicapped by the gas leaking out from the joints of this very old cast iron pipe, which is leaking. And this is a perfect kind of analog, geometrically and otherwise, for the fairy circles. And then working on this analog, I came up with this model which needs to be tested. Strangely enough, there is just one well which was drilled in Nebraska. the US, which was targeting one of these fairy circles there, and they never published the results. But this is how it might work. And again, this is a different world from what we are used to, because we are looking at really a large expression of a mega seep of gas coming to the surface. Now, this image is not coming from this signal is not coming from the hippocampus, but very similar. The story here is that and I'm not showing anything fancy here, but we do a lot of computational science too, and doing the right petrophysics and making the right assumptions. If you assume a point source for your gas in the subsurface and you let it go, it comes up to the surface in a kind of an inverse cone shape, making a fairy circle, so you have a slight topographic depression, and you do have a clear signal of that particular gas which is coming up, which is in our case is natural hydrogen. All right, let me work a little bit more with this. Let me take you to Brazil. And this big, famous fairy circle, what I show you, we call it the Mickey Mouse because it's basically a combination of 3 fairy circles, one big and then these 2 in the ears. People put monitors out there which are able to monitor the activity in the soil. And what they found that the soil, the hydrogen signal in the soil is actually showing a breathing pattern in a 24 hours pattern. And this is where the story becomes kind of complicated because we had to go in and everybody else in the business are taking new technology to put on the field, which we don't do in oil and gas. But now here we are, and then we have custom-made tools to trying to understand the gas flux in the presence of hydrogen in the soil. And this is a brave new word for us. But doing that, for example, in Austria, not very far from here, we are able to find a signal in places, this is in the Lavantal in Carinthia, where we see the same results like the Brazilians have. And there's a story behind. Of course, the story is not that simple, and then I'm not here to claim that we understand, but this is the part where we start to overlap with biology. And then some people talked about bacteria and this and that. And what we do see is a system which is modulated by a natural hydrogen system from below. And the bacteria, which loves hydrogen, the millions of bacteria, most of them look at hydrogen as caviar, and they just happily to eat it in the first 1 meter from the surface. So we have a very complicated system of having a natural signal and then some other factors like pressure, temperature, and the bacteria working in the soil to eat up the product. And then this is the part where I take you to the fault control. What folks found is that hydrogen, being so agile, doesn't like to migrate through directly the rocks and taking a long time to do it, but instead it likes to find a highway through the rocks, and the highway is provided by faults, which we do have in our business quite often. And this case study from French Pyrenees, using other gases like methane, radon, and the likes, CO2 in particular, shows that gases, not too surprisingly, like to travel along fault plains. And there are some companies who go after helium. For example, in France, they do have this new mindset. It's a very new mindset, is to drill actively or decidedly into fault plains and try to intersect that traffic of gas coming to the surface. They went after helium, which is a very good commodity these days, a critical mineral, if you will. But the same story holds for hydrogen in our view, and we are looking for conductive fault plains. And this is not what we do in oil and gas, because in oil and gas we don't like faults, because they tend to mess up our accumulations. But the brave new world we are seeing now is that we perhaps should go after faults actively. And on this cartoon I show on the right-hand side, this is the new world. It's a little bit like the geothermal field, is that everybody could have a small well of just 100 meters deep, and if you are lucky and then you are close to a fault with connected with a hydrogen kitchen, then you can drill a lot of wells, and you can farm natural hydrogen coming up from the subsurface. So totally new concept, totally new perspective on how the energy consumption and energy production could look like. And without going into details, this kind of plays, hydrogen plays, the way we approach the subsurface from energy exploration point of view looks totally different from what we are used to in the oil and gas. And almost done. And then the question, coming back to the carbon footprint and the competition with all the other hydrogens, I showed you the blue, gray, black, and pink, and whatever artificial man-made hydrogen species. Natural hydrogen, since already done and waiting for you in the subsurface, has negligible carbon footprint. It's the perfect item, the perfect commodity, if you will. Perhaps politicians don't care about that part, but what everybody cares about is the price tag. And since you don't have to pay, for example, the green hydrogen, you have to pay a lot of money for the electricity, which splits the water. You don't have to worry about those things like the blue and many other things. It has negligible or zero carbon footprint and very, very small price, less than $1 US per kilo. And that makes it very, very attractive if we could find it. We are not there yet. And in that context, this would be a lovely way to go forward and solving some of the energy problems we have, humankind, going forward. But as you can tell, they are the very, very beginning of this process. In this diagram in the lower left-hand corner, you can see that this mustardy-looking triangle-shaped thing is showing that the time where we are, and then the hope that we're going to find hydrogen more and more, and we'll be able to use it more and more for our energy needs. And my last slide, so what should be the takeaway from this presentation? We all appreciate the fact that our civilization came this far in the last century or century and a half because of oil. Okay, that this is just what it is. Everything is somehow a product of oil. And since we have a lot of folks working in America, this is an ongoing debate who got the first oil, Romania, Poland, Azerbaijan, whatever. But I picked the Titus wheel story because everybody can relate to that. And Colonel Drake, who managed to finally produce oil from 23-meter depths, 2 barrels per day. And at that point, nobody thought that this is going to be such a big deal. But it did change history for humankind, big time. And now we are at the stage that we have to dial down on this commodity because it's a nasty thing. And we have to abandon actively, decidedly, the oil and gas. But we have to have something else instead. And then the promise of hydrogen, perhaps, is that these wells drilled in Burakebugu in Mali will be the Titus wheel, but for natural hydrogen. So that's the hope, and this is what we are working on. And I hope you visually keep that in your mind. Hopefully we can come back to this 10 years later and see whether it's correct or not. Thank you.
Thank you very much. I really hope that you are right and that 10 years from now, whoever the audience will be, you will proudly present them with a lot of progress.
Thank you.
Thank you.
Thank you.
Thank you. And now for our last talk, I would like to ask Professor Aaron Chickenhauer to talk to us about his new field. He shared the Nobel Prize in Chemistry in 2004 for the discovery of ubiquitin-mediated protein degradation. He was born and educated in Haifa and Tel Aviv, and since childhood he has been interested in biological processes in health and disease, and in 2018 he opened the Chickenhauer Institute of Precision and Regenerative Medicine. And his lecture will be focused on the revolution of personalized medicine. Are we going to cure all diseases, and at what price? Please.
Thank you very much for the invitation, and happy birthday to the Academy for the next 200 years. Since I am the last of the day, and since the audience is quite broad, after some internal deliberations, I decided not to talk about my work, about what we discovered, but rather on my, I would say, hobby, which is the philosophy and history of medicine, but as relevant to our days. I will just mention very briefly what I discovered when I was a graduate student, and we are still working on very actively in the laboratory. We discovered a quality control system that degrades untowed proteins very specifically. So it degrades proteins that are aggregated, denatured, mutated, or otherwise damaged, which is very important. Think about all the neurodegenerative diseases. The hallmark of all these diseases is accumulation of aggregated proteins in the brain. It depends, of course, on the site and the type of protein, but ALS can be TDP43, SOD. All the polyQ-expanded diseases, Huntington and the androgen receptor, Alzheimer, Parkinson's, synuclein, and so on and so forth. So in this case, the system fails to remove them. Think about cancer, which is caused by oncoproteins, RAS, JUN, MYC, mutated P53. The system fails to remove them, and here we get cancer. Think about COVID-19, the discovery of the immunization. You are synthesizing a message that codes for part of the protein, Katalin Kariko and Drew Weissman, and this protein of the capsid or part of the protein is being processed to small antigens, and the processing is mediated by the ubiquitin system that we discovered. So this is one of the many functions of the system that we discovered. Based on it, there are already several very successful drugs in the market, and more are coming. But as I said, I decided not to talk about it and to talk about the revolution of personalized medicine. So this is where we are. I don't see that. Oh, here it is, yeah. So the medical school is here. Oh, well, it's the white building over this side, and it looks into the very east side of the Mediterranean, a beautiful site. The city is a very nice city. It's a place where the Baha'i people built their temple. You see part of the gardens, and they buried their founders. They smuggled it out of Iran. They were persecuted in Iran, and it became a UNESCO heritage site. Beautiful garden that goes all along the slope of the Carmel Mountain. So where do I?
Which?
Okay. Okay, so the dream of people is, of course, to remain young and beautiful forever, and also, at the same time, to be free of diseases. So the question is whether this dream is materializable. Well, history tells us that at least part of it can be materialized. Think about lifespan about 120 years ago, when the 19th century turned into the 20th century. People lived at that time about 50 years. It took one century, one single century, the magic 20th century, and we added about 30 to 35 years to our lifespan. It's an amazing achievement. We added 60% to our lifespan. In the developed countries, of course, we shall not talk about underdeveloped countries or least developed countries. The only country actually nowadays that lifespan is going down is the United States of America, unfortunately, because of violence, inability, segregation of the health system, inability to deal with pandemics, and so on and so forth, but this is a unique case. So the question is, of course, and think about history. 4,000 years ago, the Greeks and the Egyptians, they lived on the average 25 to 30 years. So it took like 4,000 years to bring it from 30 to 50, and 1 century, about 2.5% of the time, to take it from 30 to 50. So the question is whether it's going to continue this. Well, not so much so. We are still climbing up, but we are starting to get into a plateau. Predictions tell that we are able to make it to about 110, 115, and actually there are few people, the centenarians, that can cross the 100 and make it to 105, 110. Rita Levi-Montalcini, the Italian magic woman that discovered the NGF, did it to 109. I celebrated with her 100th birthday, so it was wonderful in Rome. And so there are people who cross it. And the issue is, of course, burden of mutations and collapse of systems that cannot take it for a long time. But then people say that maybe critical genes can be replaced by CRISPR and so on. But this is the prediction that we are still can climb up to about 110, 115. There are also many other issues that are related to it, and that's the economy of it. And it's, we are retiring in most countries at the age of 65, 67, 68. That still belongs to Bismarck time, when people didn't make it to this age even. So for him, it was kind of the horizon, the end of the world. And now people at this age are young and running marathon and are functional. Nevertheless, they are forced to retirement. And by forcing to retirement, they need to live on what they saved during their lives. And the savings are not sufficient to maintain them for another 20 or 30 or even 40 years. And of course, they are burdened with different diseases. So they are, well, maybe I am not politically correct, but they are constituting a heavy burden on the health providing system. And they need to be, they need caretakers and institutions and so on and so forth. So the question is, where is the balance and so on? But I will not go into it. Just wanted to highlight the issue of lifespan, which we are still extending without changing the retirement and the money-making time. Don't forget also that the market is changing. We are needing less labor in the market. We are more relying on computerized and technology, so the market is less in demand of working force. So this is also adding to this problem. So it's not just the science that we are doing in our labs that is not a problem, but that we are trying to solve, but other factors, other socio-economic factors are also coming into this problem. So you see that we are defeating diseases, we are developing antibiotics, we are developing vaccination, and so on and so forth, but that is not sufficient. Pandemics are still hitting. Cancer, we are doing a huge progress with cancer. We are actually increasing, because of the increasing lifetime of cancer patients, we are increasing the number of cancer patients that are living among us. I know the data in Israel. I will not go into it because I'm serving as the president of the Israel Cancer Society, but the number of living cancer patients is growing, and again, they are a burden on the health system, and so on and so forth. So you see that solving the problem is not, the scientific medical problem is not the only solution that will bring happiness and, relief to society. Okay, so medicine is, let's try now to concentrate a little bit. Medicine is a very complicated profession, and therapies are of all kinds. So there are engineering, of course, real engineering, metals, bolts and nuts. We are replacing joints, hip replacement, knee replacement, shoulder replacement with different metals. We are replacing valves, heart valves. We are still not able to replace real organs. We are not being able to build real organs. So for kidneys and for heart, we need transplantation, which is, of course, limited in different countries, not by technology, also by beliefs of the people, by prejudice, and so on and so forth. So this is limited. So in order to overcome part of this problem came stem cell biology. So we are able to kind of force cells to differentiate into the appropriate tissue. Well, it's not the heart that is a full mechanical pump. It's not the kidney, but maybe let's think about diabetes. All what we need to do is to generate cells that will secrete insulin in response to changing glucose levels. That is even not achieved yet. We are not there yet, but we are coming close to it. So this is called tissue engineering. So we have real engineering that is coming from universities of technology, like my institute, the Technion, and there are tissue engineering. The word engineering is not exactly the canonical engineering, but nevertheless we are trying to and the same, for example, for Parkinson. Again, many trials in order to make cells that will secrete missing dopaminergic transmitters, put them right in the brain. For insulin, by the way, you can put it underneath the skin. It doesn't matter where it senses the glucose. For Parkinson, you need to put it in the organ, in the appropriate organ, in the substantia nigra paracompacta, in order for them to function. So this is an additional complicated factor, but nevertheless, people are trying to do it. So this is one direction that medicine is going. Of course, imaging has developed in a big, big way. You know, for 8 years, from 1901, we were living with X-ray. Actually, I remember as a medical student, that's what was. You know, I studied medicine in the late 60s and early 70s, was, you know, X-ray. Now we have computerized tomography, we have MRI, we have functional MRI, we are able to see soft tissues, we are able even to look, you saw before, activities in the brain when you are being asked a question. We can distinguish between how male and females are solving mathematical problems and so on and so forth. Different organs in different areas in the brain are being activated. So we made, and imaging is not only for diagnosis and for studying and for research, it's also, there is an interventional imaging. We are putting stents in the heart by imaging. So we are doing lots of intervention by imaging. Surgery has become, you know, I'm a surgeon by training, so surgery has changed dramatically since the time that I was a resident. You know, we did really bullying surgery. I would say a butchering type of a surgery. Now we are using, you know, microsurgery. We are using robots, the Da Vinci. We are controlling the arm, which is very stable from remote. So surgery has changed a lot. So medicine is moving in parallel direction, and I will concentrate mostly on drug development, because this is what is more pertinent for us scientifically. So I will concentrate more on drug development. So let's follow drug development, and let's do it only for the 20th century. Because, you know, what happened in the 20th century, that another factor was developed, and that's communication. We are publishing, right? And our published material is being scattered all over, so people can take a paper and further develop it. This was not available in the 19th century even. So we are building on one another knowledge. I think that May-Britt cited the Prime Minister of Norway that told us this story that we all know. So we are talking only about the 20th century. So we shall talk about drug development. And the first revolution was an era of incidental areas. Drugs were developed by coincidence. People didn't mean to develop them, but somehow they came, and very successful drug. So take, for example, aspirin. Aspirin is the drug that has been sold the most by weight. Now you can buy it over the shelf. Of course, you are walking into the pharmacy, but by tons, aspirin is still being used more than any other drug. So it was discovered by whom? By the old Egyptians, 4,000 years ago. The willow barks that grew on the banks of the Nile River, they chewed them, and it was very bitter, and it alleviated pain. They reported it, the old Egyptian reported that it alleviated pain. It took almost 4,000 years, and scientists in Bayer Company, AG in Germany, purified the material, and the material was salicylic acid. And salicylic acid, why did they purify it? Out of a very strange incentive. The father of one of the workers got rheumatoid arthritis. It's an inflammation of the joints. You can see the inflamed joints in the middle in the hands. And they decided to give it, to purify the material in order to give it as a pain reliever. So they knew from the old Egyptian script that it relieves pain, but it was very bitter, so they acetylated it. It's a very simple material. It's basically acetyl salicylic acid. Amazingly, what doesn't happen to us in morphine and in corticosteroids with all the side effects, it removed the bitterness, but didn't touch the pain-relieving property. Typically, when you touch a drug, you kill it. Doesn't matter what you do. And we know it from other drugs. Here, it took away the side effect, but left the beneficial effect. And it didn't take only the pain, but it took the inflammation. So here came the first anti-inflammatory drug. Of course, with the side effect of bleeding and so on, we'll not go into it. But then people said, aha, Aspirin is anti-inflammatory, and there is a connection, early discovery, between inflammation and cancer. So it can also serve as a preventive drug for cancer. And indeed, physicians will not recommend it because of the side effect. But for families with a known family history, like colorectal carcinoma, they do recommend aspirin and other anti-inflammatory drugs that have been developed later on as a preventive drug for cancer. So aspirin initially was used also as an anti-cancer drug. And then, lo and behold, it was discovered that this magic drug can also prevent blood coagulation. Well, blood coagulation is one of the most common diseases in the Western world. It's called myocardial infarction, heart attack, when there is a clot that is being formed on an atheromatotic, on a cholesterol-laden atheromatotic lesion. This is the last event, and then the patient succumbs. People say, oh, you know, this guy was running marathons, and all of a sudden he collapsed from a heart attack. No. Yes, he did run marathon, but for decades he accumulated cholesterol in the coronary arteries that bring blood to the heart itself, and the last event was kind of a little wound on this lesion, blood coagulation, and death. And to prevent the second heart attack, people started to take aspirin, and it was very beneficial. Of course, now they have Plavix and some other drugs, but doesn't matter. But aspirin, so think about this drug that the Egyptians discovered by coincidence, anti-pain, anti-fever, anti-inflammatory, anti-cancer, anti-coagulation, magic, really magic drug, coincidence. We cannot rely on coincidence on drug discovery, and science is moving on. And the next, penicillin also was discovered by coincidence, but I will not go into it. That's the story. That's a more advanced story of Sir Alexander Fleming, a microbiologist. I will not go into it. But it was also coincidence, which also changed the world. You know, can you imagine the world without antibiotics nowadays? But I don't have time to go into it. So let's go to the next phase. The next phase was high-throughput screening. Why high-throughput screening? You know, always discoveries are somehow linked tightly to technology. We always have technology, then we make a discovery, then we say, oh, this is not enough, then another technology is coming, another discovery. It's like a cogwheel. So high-throughput screening came because we were able to grow cells in culture. On one hand, we are able to grow, you know, cancer cells, neurons, almost any type of cells we are able to eternalize. Maybe the most famous cell line, which also has an unbelievable, interesting story behind this, are the HeLa. The HeLa cells, which were taken from Henrietta Lacks, a woman that got uterine cervical carcinoma because of human papillomavirus, was operated in Johns Hopkins. The tissue was taken from her, of course, the cancer, but then it was, without her permission, was developed into a cell line that the whole world is using. So this is one partner of this wedding. The other partner is chemistry. You know, you are synthesizing organic synthetic chemistry was developed in a big way. When you are synthesizing an end product, you may cross through 10, 20, 30 steps. Think about artemisinin, the magic anti-malarial drug, 40 steps. 40 steps. So you are leaving intermediates behind that are useless. So people start off gathering these intermediates in families, and millions of compounds are there, and the pharma companies, you know it as good as I know, what they did, they did the matching. They developed robots. There is no mechanism in mind. Now there are mechanisms because of AI and AlphaFold and so on, but still now, you know, you take millions of compounds and you screen them against cells. If it's a cancer, you are screening them against cell division, and you are doing it on a grid, on 396 wells. It's all automated. We shall not go into it. And many drugs came to the market by screening. Of course, what you find initially is not a drug. It's just the very beginning. There is toxicity and safety and clinical trials and so on. But this is the beginning. And by screening large libraries, one of the drugs are statins, multi-billion-dollar selling drug. It's an inhibitor of the first enzyme in the biosynthetic pathway of cholesterol, the HMG-CoA reductase. Was discovered by Akira Endo, a pharmacist. You see him here. In a pharma company in Japan, he screened a library of about 100,000 natural compounds Japanese plants. So all these libraries are kind of concentrated or focusing or derived from one source. So different plants, and he found one that stopped cholesterol synthesis, again in cells that are making a lot of cholesterol. The company sold it to Park Davis, an American company, and statin is a history. It's a multi-billion-dollar selling drug. The last year's sales were $40 billion, and it's really prevent cholesterol precipitation in the coronary artery for people after a myocardial infarction. But because it has to do with cholesterol, people tried it on other diseases that has to do with cholesterol and with lipids. Alzheimer's disease, there are some signs of prevention. I don't want to go into it. It's complicated. And for prostate cancer, it's already well established. So you see that all of a sudden, drugs start to get repurposed. And we'll talk about repurposing later on, because this is a new movement in development of drugs. So this is this part. We are moving to the third revolution. And the third revolution is where we are now, and we call it the revolution of personalized medicine. It's not a revolution, it's a response to a technology. That's all what it is, and we are already, I wouldn't say in the middle of it, I would say rather in the beginning of it, but it all comes to these two gentlemen, Francis Crick and Jim Watson. Jim is still alive. I came to know both of them. Francis died of cancer several good years ago. And it's the double helix, which is the foundation of all what we know nowadays. In my opinion, this is the biggest discovery in biology in the 20th and 21st century. It's really an amazing discovery because it gave an answer to all what we know basically about modern biology, but that's my opinion. And the new drugs are going to be based on the DNA. And so basically, if you think about it, we are moving from what I call pajama type of drugs, or of drug development, to suit tailor-made. What is pajama? Pajama is something that you don't care much unless you are really a little bit crazy. You go to the shop, there are 2-3 sizes, small, medium, and large. You buy one, and the differences can be adapted by the rubber band. For tailor suit, you are going to the tailor and do it. And what this is all about? It's all about what we see here. I think that what physicians noted, that different patients respond differently to the same very drug at the same very disease. But the same very disease now, as we understand it, should be put in quotes. There are no people in the world, 2 people in this planet that are identical to one another. None. Even not identical twin that came from the same very sperm and from the same very egg. Because there is epigenetics. We forgot about it, and once we are out, we are going different ways, different diet, different climates, and our genome is changing as we grow. Of course, different people are different anyway, but even identical twins that we thought are identical are not identical. Therefore, there is no wonder that the disease, what we thought is the same disease, behaves differently. I remember when I was a surgeon, breast tumor was a breast tumor. What we did a biopsy. The pathologist, by looking at it in a very primitive instrument called light microscope, told us this is a tumor, and we tried to identify whether there are metastases. If there were no metastases, we treated the woman by a very mutilating surgery, taking the breast out, then chemotherapy, radiotherapy. If there are metastases, it was a little bit more complicated, and so on. But we didn't go into understanding the mechanism of the disease, what underlies it. We thought a tumor is a tumor, and Alzheimer is Alzheimer, and Parkinson is Parkinson, and heart attack is heart attack. We define diseases by symptoms and by phenomenology rather than by mechanism. This was a conceptual, I wouldn't say mistake. It was not a stupidity or something. It was the state of art of understanding at that time. And now it's changing again because of the human genome. And I think that the second achievement of the human genome was the ability to sequence the human genome individually in a very speedy way and pricey reasonably pricey way. So, you know, the first human genome took almost $1 billion in 7 years, and it was only partial and so on and so forth. Now we can do it almost at no time and at no price. We shall not go into how the prices are going down, computer sciences or software that was developed, and that allowed us to stratify diseases according to their cause, as simple as we are living now in this era, but you know, until 20 years ago, it was not clear to us at all. I always, because I like to drive, I always compare it to a car that doesn't turn on in the morning. This is the name of the disease. You cannot turn your car on. But it can be the battery is empty because you left your lights on. The gas tank is empty because you forgot to fill it. The oil tank is empty because you rode a bumpy road and a stone drilled a hole in your oil tank. It can be that there was a rain and you have a rusty engine cover and your sparks are wet. So if you repair one problem, the car will not turn on. So this was exactly what we did. We defined diseases by phenomenology. The car doesn't turn on. A woman has a tumor, a man has a tumor, and so on and so forth. And this all changed now. Well, we are in the process of changing it now, and we start to stratify diseases, and we are moving into personalized medicine. I think that the first hint came to breast cancer, when people started to understand the stratification. And you can see 2 biopsies on the left side, taken from 2 women. You can already tell the difference, but the main difference is the color. The color is artificial. On the right one, the two samples, the 2 biopsies were stained with the same antibody against a mutated estrogen receptor, but then one on the left side didn't catch the antibody, and the one on the right side did catch the antibody. So the woman on the right side has a mutation in the estrogen receptor, and the woman on the left side has something else. So the woman on the right side can be treated with tamoxifen, which counteracts the activity of the mutated estrogen receptor, while the woman of the left side, if we'll treat her with tamoxifen, we shall waste money, but mostly hope, and she will continue to deteriorate. And until now, we treated the 2 women with the same treatment, not with tamoxifen, with mutilating procedure that, it's like shooting a fly on the wall with a cannon. That's what we did. And now we are doing it with very gentle forceps. We are really doing a dissection of the underlying reason that causes the disease. And the woman on the left side may have a problem with a mutation in the HER2, in the EGF receptor. So she should be treated with Herceptin. A third woman might have a problem with the progesterone receptor. And a fourth one, unfortunately, will be triple negative, which is the worst. And it's the worst because we don't know why it's there. But we'll soon know why it's there. And there will be the prediction that there will be about 15 different breast cancer, all related to different mutations and different alterations, and they will all need individual, distinct, and different treatment. Now, we are on the road. So what we are doing with the one that we don't know, we are, of course, mapping the mutation. And now with AlphaFold, we don't have even to crystallize the protein. We can predict the structure, more or less, and then we can use computational chemistry in order to dock, to synthesize, to plan, to design a small molecule that will dock into the active site and will inhibit it. Of course, we shall need, I don't know if animal experiments, but clinical trials, I believe that we still need, because at the end we are treating people, but that's up. The FDA now is trying to push back on animal experiment. We heard this morning a wonderful lecture on tissues on chips, and we can do a lot of toxicity on just on organoid, on small wells in which cells are growing or organoid, and measure the entire omics in the output fluid. So this was another advancement of medicine that may abrogate the need for animal experiments, but I will not go into it. Clinical trials, but it will shorten the way in a big way to develop drugs, and mostly it will reduce the price for about $10 billion per drug. That's the average now, to about, I wouldn't say nothing, but to a much less than that. So I think that this revolution will coalesce into a much more efficient, time-wise and price-wise, development of novel drugs. So you see what's going on. Okay, of course, it might solve also the problem of side effects, again, because when we are treating different patients, not only that they respond differently, but the response can be, yes, we respond to the drug, we respond partially, we don't respond at all, we respond with mild side effect, we respond with severe side effect, we have only side effect. And the side effects are also unpredictable. I remember in the emergency room as a young doctor, what I asked the patients when they got wounded, whether they know about sensitivity to penicillin. That's all what we knew. We knew nothing about sensitivity, but people are basically sensitive to every compound. Yesterday during dinner I heard that people can be sensitive even to alcohol. I don't know whether this is a good sensitivity or bad sensitivity, but they are sensitive to alcohol as well. It's a small molecule. I cannot imagine that it elicits histamine release and from IgE activation, but let it be. So it will solve also the problem of sensitivity, and that's the reason we are different. We are men and women, fat and thin, carrying different genetic heritage, exposed to different climate, different diets. Not two people that walk on the face of this planet are identical to one another, and therefore we cannot expect the disease, of course, any disease, to be identical course, behavior in any people, except that now we are developing tools along with AI and big data sets in order to predict how the drug is going to act on a patient. Okay, so let me now, so of course it's the human genome, and of course we are developing banks, we are sequencing, not we, I mean the community of each disease, about 5,000 to 10,000, that bringing us to almost a plateau of almost all reasons. There will be some rare reasons behind that are very rare that we shall not identify for each disease. But that's about it. And of course, we are building in the world what we call the banks of sequences in order to identify the different mutations that are causing different diseases. We are talking just on not genetic diseases, but diseases that are caused by mutation. There is a difference. Genetic diseases are inherited diseases. There are many diseases that are acquired. They are still affecting the DNA, of course. So we are limiting ourselves, but to quite large diseases. We are trying even to do it on psychiatric diseases, but that became very complicated because, we are thinking in genetics of monogenic diseases. Many diseases, metabolic diseases, heart diseases, and so on, but mostly psychiatric diseases are polygenic diseases. And then we are really walking in the dark in many ways. Therefore, autism really popped out as a spectrum of diseases. It's not a disease as we thought before, but it's rather a spectrum. 20 years ago, a system was introduced to our testing arrays, which is GWAS, gene-wide association. I just read a review on GWAS. It didn't bring much, I would say, benefits in order to this. So we know connections and association, but drugs were not developed based on GWAS. So the situation with GWAS is a little bit kind of iffy, but maybe in the future it will change. So we are still, even with all these developments, are rather difficult, rather walking in the dark. Of course, based on genomics, further omics were developed and they were described today. So we are moving from genomic to transcriptomic to metabolomics to proteomics and then to metabolomics. So there are 4 big omics around and we can do them all. We can profile a patient very precisely. But again, think about it. What we do is true for today. Tomorrow it can change. So here comes again the fact that we are not isolated in the lab. Think about insurance company that will do, okay, they will force us to do, omics in order to insure us, but the omics of today might not be the omics of tomorrow because it may change. So this also puts a burden, and the insurance company will discuss it, may prefer that you'll get sick rather than then the whole population will have periodical test every year in order to prevent one from getting this. They prefer to treat one cancer patient than, identifying a candidate for cancer among periodical tests that are being carried out all the time. So you see the balance is all the time. We have to turn our head also to the society and to the price and to what it takes. So these are the problems here. I will not go into all of them, but I still want to highlight two of them in our approach to personalized medicine. Of course, I mentioned the polygenic diseases, the GWAS that are still very burdening, but I would like to look into the last two ones, the 6 and 7. 6 is end of blockbuster drugs. There is a constant fight with the drug companies and with the pricing. There were few chemotherapeutic drugs for AML, for CML, and they are still there, that we are using for solid tumors, for blood tumors, and so on. For cholesterol, only statins were in the market. They changed, but they were only statins. Now we have other ones. We have anti-CKS9 and so on and so forth. We have CRISPR-Cas in order to mitigate the activity of some genes. So all these were developed by the companies, enjoyed blockbuster. So drugs were sold for billions and billions of dollars, and patients took them even if it didn't help them. Let's say that a statin for one patient reduced the cholesterol by 10%. The patient said 10%, it's good and better than 0%. So let's take it. Let's say that he suffered a little bit from muscle ache and due to the statins. He said, okay, maybe it's worth to pay the price than to have my arteries occluded with cholesterol. So, drugs were used and companies made billions of dollars. Now, if we are going to slice the diseases, and breast cancer will have 13 or 10, and cholesterol already have 4 or 5 reasons for it, statin market is plummeting. And other drugs are coming, and the companies really, I would say, don't like it, and it's also competition. One company has a patent, so the other one cannot enter the market for so much time, and so on and so forth. So this is the blockbuster era is going to begin. Maybe the companies are going to be damaged, but the patients are going to benefit. But then comes a very big issue, and the big issue is the bioethical problems that are attached to it. And that has to do with what we want to, what is a disease, and what we want to know about ourselves. How many people will volunteer in this audience that will sequence their genome? Beside the fact that that there is a danger of hacking into the information and so on. If I have a criminal mind, I can walk into this hall and just wipe the armchairs with alcohol before you enter, and then after you leave, and I'll find the sequence of each and every one of you because you left your hand somehow on the armchair. So this is one example, but people are hacking into computers of hospital, and then they are asking for ransom from patients. Hey, I know what you have, and I'll bring you some examples. But let's don't talk about crimes. Let's talk about real issues, about what we really want to know and what other agents would like to know about us beyond asking us questions whether my mother was a little bit kooky and whether my grandmom died of some disease that I don't remember and so on. Think about insurance policies, what we are being asked today and what they can really ask in order to know about, the demand can come even from customers. Let's say that there are two customers. One have a history of diseases in the family, the other one doesn't have. Why should I share the policy with somebody and pay 10,000, euros or whatever a year, and he will pay the same if he can pay 19,000 and I can pay only 1,000 because my family history and my genome tells them that so it can come also from bottom, not only from top, this demand. So you see how these health issues have huge implications on our current and certainly even short-term future life. Genetic privacy, we talked about it. In our hospital, they are already doing, in every modern hospital, they are doing now not full genome sequencing, but for cancer patients, we are looking for 300 oncogenes, and they are all going into the database. And all what I'm showing you is not taken even not from the New York Times, it's all from Nature. The genetic privacy, and be prepared for the big genome leak, ransom, and so on and so forth. But again, let's leave this one aside. Think about Angelina Jolie. that her mother died of breast cancer, and her aunt, the sister of the mother, died of ovarian cancer, and she suspected that she's carrying BRCA1. And indeed, she was found as a carrier, and she did these 2 preventive operations. Now we are not doing it in surgery. We are following the patient. We are doing it only on the ovaries, which is really fatal, because when the tumor is discovered, it's in all cases, basically, it's too late. Breast can be tested, so some women prefer not to do it, and the surgeons and the breast oncologists accept it, but they are under a tight follow-up. There's 40% of developing breast, 50% breast or ovarian cancer, which is a ticking bomb. I mean, this is a ticking bomb that can explode. And what she did, she removed the ticking bomb from her body, and she really declared it in order to encourage other women to do the same if they have a family history. It's very interesting. Here again, you know, we are sitting, this was Mary Claire King who discovered BRCA1, great achievement. It's a cause, a mutated BRCA1 as a cause for breast cancer. By the way, it's a ubiquitin ligase. It's part of the system that we discovered, but nevertheless. But again, you know, this is not sufficient. I think about Israel and the United States. The ratio per capita of women that will go to the doctor with a family story per capita in the United States is 10-1 in Israel. Why? Because in Israel we have a lot of religious people, and they believe in what Americans have on their quarter, in God we trust. Okay, they trust in God, the Americans trust less in God. They go to the doctor, the Israelis trust, the religious women trust in God, and this is the result. 10-1 ratio per capita of going to the doctor. Amazing. You have a discovery, you have a ticking bomb, everything is known. The Israel Cancer Society advertises it everywhere in the world. That's what we got. So that's it. But this just shows you the complexity. Medicine is not a laboratory. We are dealing with people. And people are strange creatures, complicated and strange creatures. Okay, so let's now, in the very few last minutes, go to the implications of definitions in medical education. Since I'm in a medical school, medical education is very much close to my heart. The 3 pillars of medicine are disease, patient, and treatment. And they are shaking. They are going to disappear in lieu of this revolution. So think about, you know, we think personalized medicine, we shall sequence, find the mutation in RAS, do this, do that. No, the implications are huge. So a disease, you know, you can see on the left side the old disease, and you can see on the right side the modern disease, which is the very sophisticated diagnosis. By the way, in Milan, I just read in Science magazine, there was discovered under, it's called Ospedale Maggiore, a big hospital that was built by the government, by the rulers of Milan, that hospitalized the poor people, not the church, the secular management, and they found now a huge graveyard of patients who died with thousands of remains of bones that they are now sequencing in order to get a picture of the diseases that were prevalent and common in North Italy. At that time, it was just, look under Ospedale Maggiore, beautiful story, archaeological story. So the disease, think about biomarkers to Alzheimer. You know, I go to the hospital for a chest pain. They tell me, Aaron, we have good news for you and bad news for you. The good news that you don't have heart attack, but the bad news that you have an APO4E. Okay, what do I do with it? Go to my wife, go to my employer, go to whom I go with a high chance of developing, and you have the 2 alleles, not one, the 2 of them. So what do you do with it? So, you know, typically Alzheimer is discovered, we are trying to get biomarkers, but it's discovered by symptoms of the patient. But what about early discovery? That's the aim of medicine, to prevent diseases and to treat them when they are discovered early before severe symptoms evolve and erupt. Think about newly developed blood tests can detect cancer 4 years before conventional method. What does it mean? That this patient is sick or is he healthy? Now, what does it mean has a very broad meaning. What does it mean for the family? What does it mean for the children that may carry the same mutation? What does it mean for the employer? What does it mean for the health insurance? You know, we are not isolated. We are living in, we are connected with ropes to entire cohort of agents that we are dependent on, and they may have their own decisions that are not totally dependent on us. So think about it, about what early detection can mean. When you are sick, the society already accepts that you are sick. Go to the hospital, you go to the doctor. Some diseases are still stigmatized, but that's accepted. But when you are healthy, what does it mean that you are carrying a mutated P53 or a mutated RAS? What does it mean? So that's the disease. The patient. Why the patient have to be 50 years old chest patient? It can be an egg, it can be a sperm, it can be an early embryo. Now think about experiments, what we are going to do with it. Think about church, think about society, think about prejudice, think about cost. But the patient can be, we are doing now it in IVF. Think about in vitro fertilization. We do test embryos for health and for basic diseases. So we are there already. So the patient, at least at the very beginning, of course, as I said, it's going to change a long time, is going to be very different from the canonical patient that we are seeing in the ER when we are on call in the evening or during the day. And the treatment. Treatment can be genetic editing, right? Well, people tried it. Look at this Chinese guy that declared a few years ago in a conference in Hong Kong on trying to prevent AIDS from two twins. Their names are still hidden, but they were called Lulu and Nana. And, you know, what did he do? Well, in this case, it was a crime because at that time there was still off-target effect. He wanted to inactivate this receptor that is the entry receptor for the virus. So he was successful in one kid and not in the other. And of course, it was very dangerous. The Chinese kicked him to jail because they were under this avalanche of attack from the scientific community. But I think that initially they supported him with the hope that he may somehow shake the hands of the King of Sweden. I don't know what really was behind, but that was maybe the thinking. This was very preliminary in this case, but people tried it, and half successfully, so-called, we still don't know. The reason was that the mother was infected with AIDS, and and of course there are now methods not only for cesarean section, but even for vaginal delivery, and the kids are born clean of the disease. So medicine knows how to treat it without this, but he tried it, but it's coming. It is coming for sickle cell anemia, mostly for monogenic diseases. In the black people in America, there is single mutation for monogenic diseases and so on. We are doing it, and I think that we are on the verge, not using the same CRISPR-Cas that was developed by Emmanuel Charpentier and Jennifer Doudna, more sophisticated method, but nevertheless, it's coming into use. So you see how the disease, the patient, and the treatment are changing in front of our eyes from the canonical one, which is for the good, but with huge ethical issues behind. I'm not talking about the pricing, with the huge ethical issues behind. So I think I'll leave you with that. I hope it was okay for the end of the day. You're all hungry. I will release you 10 minutes ahead of time. And again, happy birthday to the Academy, and thank you very much. Oh, thank you very much.
Thank you very much for coming and for an amazing lecture.
Thank you very much.